Hello everyone, my name is Osman Hamid and today I will be presenting the path of a protein, of a specific protein in my case. For this protein, the first step is beginning at chromosome 7 in the nucleus of a cell within the lining of the intestines. RNA polymerase attaches to the gene, divides and breaks apart each code of DNA. Transcription occurs to one side of the DNA and the other side becomes mRNA. The new mRNA leaves the nucleus from nucleic pore and goes into the cytoplasm where it finds a ribosome from a rough ER. Here, the ribosome initiates the translation portion of the process by reading the mRNA that then lets the three-coded transfer RNA create the amino acid chain. Once all the amino acids have been added, the chain of amino acids are released and a transport vesicle leads it to the Golgi body. Here in the Golgi body, the folding process begins that turns the product into a protein. After the assembly of the protein is finished, the secretory vesicle leads it to the microvilli populated side of the cell. Here, the protein attaches itself to the outside of a microvilli. On this slide, it's my protein model. On the left is an image of like a more of a bird's eye view, and on the right is more of a side view. If you look on the left, you can notice the Golgi body, the rough ER, the nucleus on the bottom left with the nucleolus as a pink ball. Um, there are orange ribosomes around the ER, and I've labeled each step with a sticky note. Um, you probably have noticed the shape of the cell, which is a very unique shape, and this is due to its position in the body, its place in the body. This is, as I mentioned before, part of the lining of the intestines, and the top part is actually the microvilli coming out of the cell. And later on in the presentation, I'll explain more about the purpose of these microvilli for the cell. Now moving on to the protein function. The cell this specific protein is made in is a cell that is part of the lining of the intestinal tract. This cell's function is primarily to absorb as many nutrients from the passing by food as possible. This non-ordinary cell is called a brush border cell. The difference between a normal and brush border cell is that the brush border cell has one side of it made specifically for the purpose of absorbing the most amount of nutrients from food. This side of the cell contains minute cylindrical processes called microvilli that in return significantly increase the surface area of that side. By increasing the surface area of the cell, nutrients have much more opportunity to enter the cell. Due to the cell's primary goal of collecting nutrients, the cell's organelles and exterior are specialized for this task. One of the parts of the cell that make its job impossible is a Maltase-glucoamylase protein. This protein's location is attached to the microvilli on the exterior of the cell. This area is the perfect place for the protein to function due to its position in relation to the starch that may come from that side of the cell. This protein, alongside the sucrase isomaltase and alpha amylase protein, work in synergy to digest the complete range of dietary starches. This brush border maltase glucomylase protein serves as the final and crucial step for intestinal digestion of dietary starch for the body. As you can see in the top right, I've included an image of this particular cell. If you look in the top, where I've included an arrow on the left side of the image, you can see the microvilli that I'm talking about. This significantly increases the surface area of the cell in that region. Now on to the mutation and diseases that this protein can have. There are no real actual accounts of gene mutation that have happened with this protein. Um, and the main error really associated with maltase-glucomylase protein is the maltase-glucomylase deficiency. Um, this deficiency is actually very rare and only several accounts have ever been recorded. Symptoms are similar to those seen in CSID, which is a disease similar to a protein that's similar to the maltase-glucomylase. And diagnosis requires a demonstration of reduced glucomylase activity in the setting of normal small bowel histology. Affected infants usually develop symptoms soon after they first ingest sucrose, and symptoms usually include explosive or watery diarrhea, resulting in abnormal levels of fluidity within the body or water within the body, and abdominal swelling or abdominal discomfort or pain, and in many cases malnutrition to the infant or even person, which and that's due to the loss of ability of absorbing that specific nutrient, which is starch. And this is my works cited. Thank you so much for listening. I hope you enjoyed.